Okay, good morning. Thank you, the chair. First, I would like to thank the organizer for giving me the opportunity to present of the work for the 3K rice project. But actually, unfortunately, until now, we don't have much data. This is a huge effort. I only received a few slides the day before yesterday. But what I'm going to talk today is try to give you a different view about the representing different group on what is we try to do. This the 3K project was a very important component for the Green Soup Rice project, funded jointly by the Chinese government and the Gates Foundation. So on behalf of the group, including the CAS, Chinese Academy of Agricultural Sciences, International Rice Research Institute, and the Beijing Genomic Institute. So we initiated the project. So from the previous speakers, we know there's tremendous genetic diversity within the Oryza genera. But you can see here, we know the domestication has reduced tremendously of the genetic diversity within the primary gene pool of rice. However, the breeders worldwide are still focusing on using the diversity within the primary gene pool of rice with a very limited effort in exploring diversity in the wild gene pool, because we know the difficulty in using the wild species in breeding program. So now if we look at the current status of the germplasm use in the worldwide, we know the collection and conservation is largely done. Characterization of the germplasm resource is very superficial because mostly at a phenotypic level, which is relevant to a specific environment, and also we know the molecular assessment using DNA markers or using resequencing revealed tremendous diversity within the primary gene pool of rice, but linking to this molecular diversity into the phenotypic diversity, into the trait and to gene function is the huge challenge. And in the breeding evaluation of this germplasmization is trying to identify some of the lines with a desirable phenotype to be used as a donors. But we found that there are very few of these germplasmizations had the so-called desirable phenotype. So we still do not understand how much genetic variation for the trait important to breeders in the primary gene pool of rice. In terms of utilization, we know there's an unfortunate fact that very small portion of the germplasmization in gene banks has been utilized in breeding programs. The reason have several reasons. We know conventional breeding has a limitation on using many parents in their breeding program. We also know the outstanding commercial genotypes are commonly destroyed if you are using the unimproved exotic germplasm in your crossing scheme. We also know that a slow but consistent improvement can be made within a narrow genetic base of inbreeding program. So most importantly, in breeding program, selection of the parental line in breeding is largely based on the phenotype. So in this project, there is a concept trying to establish core collection of rice germplasm. This slide shows, you can see only 5% of the sample, a good sample in the germplasm that can represent 90% of total diversity within the gene pool. That's tremendous. So only 0.5 or 1% of the sample can represent about 70% of the total diversity. This number of the sessions representative that we call the core collection or mini core collection, it is manageable, can be utilized directly by breeders. And this is the reason why we want to take advantage of the sequencing and try to help the breeders to using the modern tools. So the goal of the 3K rice project is try to reveal the details of the genome of the rice core collection in the worldwide that will represent approximately 90% of total diversity within the primary gene pool, with the ultimate goal to turn rice gene bank into a permanent database and information repertoire that ready to use of genetic resources accessible to worldwide research community and for developing novel breeding strategies for rice improvement. This is our goal. So to achieve this, we have a good breeding strategy, try to sample to create this core collection. This is the number of sessions, the total number of sessions. We selected 3,700 of sessions from 81 countries. That largely representing the total, we currently knowledge about the genetic diversity within the primary gene pool. This is including 35% from the Southeast Asia, 21% from China, 26% from South Asia, and others from different countries. These have a very good representation of the geographical distribution of the species. Now we have finished the sequencing. This table summarizes the current status of the sequencing. From Erie, we have 3,000 sessions. From China, from Cas provide another 600. So we have these numbers. So we qualified the data from 2,500 together. We have 3,100 lines have a good quality of data. So sequence sample is this is about we have this number of lines about 3,000. We have average coverage, genome coverage about 4x or more. We have about the average the sequencing depth is 13x. So the total produced, the reads is 205 gig base, and then the produced basis is 17T. This is a huge database. So this is summarized what about the situation. We have a big portion of the lines with about more than 2,300 lines with it's about 10x or deeper data. So these slides show the mapping result when we try to map all this sequence data on the reference genome of Nippon barrier. Then we found most lines would give you 80% of the, more than 80% of the map rate. But we still find a significant portion of the genome that have a low, relatively low portion of the mapped, that can be mapped on the reference genome, which means probably based on a single genome that may not really allow us to reveal the total gene content, the diversity within the primary gene pool. And there's still going on activity to try to understand what's the nature. And these slides show a preliminary final genetic tree. This is actually from something 1,600 of lines, which is unfinished tree. But again, this tree also show the typical population structure of the cultivated rice, including the most in the Indicas represents the largest group. Japonica, including the tropic Japonica and the temperate Japonica, well-separated groups. We also see the Aus group. This is clearly as identical structure revealed by many previous talks in Dr. Han Bing's group. These slides show the SNP polymorphism site that revealed in this study. We were able to detect a total of about 20 million total SNPs across the rice genome in the 3,000 sessions. You can see they are really, on average, most of which have more than 1 million, or about the highest in chromosome 1 is close to more than 2 million SNPs on each chromosome. The number of the SNP is correlated, somehow correlated to chromosome length in physical length. And this, you can see the SNP distribution along the cross, another presentation of the SNP distribution on the rice chromosome. So they are really largely well distributed around the 500,000 number. So talking about the power or SNP calling, we found the most SNP detected are the we call the frequent ones that have a frequency more than 5% in the sequence sessions. This number of SNPs account about 75%, 76% of the total numbers. That means a majority of the SNPs have a high level of differentiating power in detecting the differences among or between the different sessions that can be utilized in the future breeding program. But we do find a big portion of the rare alleles, the SNPs that are showing to have a low frequency. And this probably, we need to understand, probably associated with the with the breeding process. That some of them associated with the breeding process. Then these slides show the distribution of detected indels per entry. You can see, if you look at the number of indels so that you see most of these indels have a frequency per session. You have a large number of indels, average from 150K to about 375K per session. This is a huge number of indels detected in the different. But again, I say this is a very, very preliminary description of this project. The purpose on behalf of the group, we just make announcement, we want to make all the sequence data publicly available soon, immediately after we have the first publications. We'll also make sure to the group that all the seeds of the sequenced material will be made available. You can contact Dr. Ken McNally of Erie for the sessions from Erie, and you can contact me for the Chinese sessions for the availability of the seed. I would like to take this opportunity to call for the international effort to take a good advantage of this sequence data for deep mining and analyzing this sequence data and establish that will contribute eventually our purpose to establish the permanent rice genome functional database. Now, you're probably wondering why we want to spend so much. This is more than something like a $4 million investment in generating such a huge data. Then what is the purpose of this, what we want to use this, the data? So as I mentioned, this project is a very important component of the Green Super Rice Project Phase 2, then that is developing varieties that for the many countries in developing countries. The definition of the green super rice is already defined by Dr. Chifa Zhang, that is the variety that can produce high and stable yield under less input. So now I'm trying to show the concept how we try to linking the genome resources, and the functional genomics with our breeding activities. This is a breeding strategy we have been utilized in the past 15 years. It's integrating the mini core collection from worldwide, the minimum core collection of the Chinese rice germplasm intra-elite rice background. 46 elite varieties, best varieties in China, crossed with something 600 lines, and create, we call it by background breeding, we develop large sets of these what called trait-specific integration line sets in the elite background. Then we hope to develop SNP and sequencing information to allow us for the largest scale gene discovery, that because this breeding process will produce a lot of varieties, and that information can be directly relevant to our breeding program. And you can see, I don't have to show the outcome. This is a huge effort for the last 15 years. We have developed a tremendous, this just gives you a single set developed in a few years at Erie, using the same strategy as large as we can turn a few elite lines into thousands of the, we call trait-specific integration lines. Each carry a different trait drought tolerance, different, we call the green traits for the integration lines. These slides give you a summarized results from this integration breeding. What we found is there are tremendous amount of the genetic diversity for almost any complex trait in the primary gene pool of rice we screened, most of which is hidden in the exotic germplasm sessions. What does it mean is you cannot predict a line or a session whether it has good genes for the trait you are interested based on their phenotype. Second, we found the selection of parental lines for breeding based on phenotype practiced by the most breeders is a poor way in exploiting this hidden diversity. Now I'm trying to challenge this group is what is the genetic and molecular basis of this hidden diversity for complex trait that remains poorly understood. So this is a future breeding we will try to do. We try to develop two large platforms, which is a GSR material platform. It consists of large number of integration lines that will be phenotyped, genotyped, and the same. And this breeding 3K project will develop, we call the green supervised information platform that will support the linking the DNA sequence polymorphism with the genes trait we discovered from the breeding program that eventually allow us to find a large number of genes, finally providing the tools and information for us to develop new and novel breeding strategies for developing this green super rice. This is a whole effort. So now I'm trying to give you an example how we're going to use what does it mean, this hidden diversity, and what does it mean, what is the breeding, what does it mean, the hidden diversity. This is HHZ is a very mega variety that are grown currently in 2 million hectares in South and Central China. It has tested in more than 17 countries with wide adaptability, with very high yield, superior grain quality, and yield potential. So now we cross this line with 16 donors from diverse of a region. And you can see this is representing a typical breeding program. You cross with the ED lines with many donors. Okay? Differences in this case is the donor has not been selected for any trait. It's just a set with representing diversity. You can see from different countries and different origin, and including different subspecies. This is a integration breeding process we developed with this. This is the first set of it we selected for yield, drought tolerance, salinity tolerance, submergence tolerance, etc. That creates a number of integration lines with a specific trait. Then we cross-protein test for multiple traits that give you many lines that eventually led to, we call it about 500 lines to be genotyped and for testing for all kind of traits. And that we are leading to new varieties in the preliminary yield trial and eventually for varieties. At the same time, we are also using this integration line for gene discovery. This is another set. The second 8 population were done within 2 and 3 years. We can convert it a single variety into thousands of these what called trait-specific integral lines with this kind of many traits. This slide shows, you can see from the second set of the 8 populations, we were able to develop these 800 lines with improved trait for drought tolerance and multiple improved trait. You can see with drought tolerance plus good yield on the low input, drought tolerance plus salinity, drought tolerance plus submergence, drought tolerance high yield, low input performance plus salinity, etc. Multiple trait is 3 traits. You can see this is the line already entered the preliminary yield trial. This is the number already entered the multi-locational yield trial, and is going to be released to farmers very soon. So you can imagine, remember this set of the material we call all are transgressive segregants because they are better for this trait than their both parents. So this is what I call the hidden diversity, because in both parents, they are not as good as any of these integration lines for the complex trait. And you can see just the select part of the lines in the preliminary yield trials for the grain quality, you can see the diversity of the grain quality traits. You can imagine that the genetic diversity can be easily integrated from primary gene pool in the elite background. So the greatest challenge is, while these integration lines provide the useful, it's already breeding process, useful material for functional genomic research for complex trait, but our results suggest association mapping based on germplasmization is unable to detect this rich source of hidden diversity because the parental lines do not differ very much for most of the traits we measured. In other words, if we do not see the diversity, the phenotypic variance, even you have tremendous diversity at the genome level, you are unable to detect the genes associated with this complex phenotype. So now I'm trying to give you an example using the integration lines for the large-scale gene discovery, how we can understand the complex genetics, of this complex of the trait, I'm using a highly heritable trait for the resistant to bacterial blight resistance. You can see this is the 8 population. From first group, the number of lines was selected. Actually, remember they are selected for submergent abiotic stress and not selected for bacterial blight resistance. And this is the process of breeding, I just repeat, but I'm emphasizing the selected integration lines were phenotyped for multiple traits, including the resistant to bacterial blight and blast, etc. Okay, we have the phenotypic data. Now is the result, try to understand what is contained. This slide shows the responses of these 9 parental lines to the 15 bacterial blight races from tropical and from China. You can see they are showing the diverse of the resistance spectrum to these races, ranging from completely susceptible of some of the parents, highly susceptible to all the races, to some of the lines with resistant to 7 of the 15 races. Okay, so this is no high level we call it immune of this type of reaction was observed in any of the parents. This is a progeny from part of the 131 integration line from 2 populations. These are interesting populations. You can see we have a large number of progeny showing high level of resistance. HR means immune, virtually hypersensitive reactions. The other types we call resistance, the red one. We also found some we caught lines, susceptible line from the population where both parents are highly resistant. Okay, you see this high level of resistance to all the races. You can also see the susceptibility generated by recombination. So this certainly, because both parents are not, we see some lines resistant to all the 15 races. We call this probably resistance caused by complementarity. This will give you another population. There are different populations from there in terms of, you see, this is another population. From these 2 populations, you can see from these populations, both parents are highly resistant, but we found a big number of progeny are highly susceptible to specific races. That is the susceptibility caused by recombination. This give you example. This is there's a recipient, this is a donor, you can see, and from progeny we find some lines are highly immune to all of the 15 races. This portion of lines are showing the immunity to the pathogen. And if we look carefully of the reaction of different lines, we can find that there are 4 types. Immunes to all the races, highly resistant, the resistant with the lesion length shorter than 4 or 3 centimeters. We have moderately resistant or susceptible lines. You have the lesion length of this much, about 4 to 7 centimeters. We find many highly susceptible reactions. This is a typical, using just based on the phenotypical reaction of the HHC lines, we could easily classify these 15 races into 2 major groups. One is called weak virulence strains or races, and the other group, including most of the lines, we call the strong or high virulent group. Now, I'm using another few minutes to demonstrate how we're going to use the sequence-based information for the genetic mapping for the resistant genes to the bacterial blight. We know up to date, we already added 35 loci reported for the bacterial blight resistant today. This is based on the sequence information of the parent lines. We can see the SMP polymorphism between the lines. Even all the 8 lines all belong to the same indica group. But this sequence information provides a tremendous SMP allow us to detect any integration in each of the lines. We also sequence the 500 lines. This is the integration pattern identified in the 8 populations, the integration lines from the 8 populations. We divide into the whole sequence information into about 38 bins with about each bin of a size of 50 kb to 200 kb. Then we perform the association, the genetic mapping. We can find large number of resistant loci associated with resistant to specific races in the populations. This is a population detected into 6 populations against the race 1. We use only 4 race example because this data again is incomplete. And this we're using 2 weak violent races example here. We found this number of loci, resistant loci associated with the across genome. And we saw another 2 violent races against. We find more resistant loci across the rice genome. Then you can see another very violent race 7 from 5 populations. So you can see the number of resistant loci identified. This shows on the chromosome 1, 2, 4 the distribution of the resistant loci across the rice genome. You see these boxes actually representing the loci associated with the hyper-sensitive or immune reaction detected into 2 populations. This is another set of the loci detected across from chromosome 5 to chromosome 10 and across the rice genome. And is also this on the chromosome 11 and 2. We already know this big chromosome contains a large number of already mapped known genes, including XA21, XA26, so many other genes in this cluster. Again, we find a large number of loci clustered in this group, and you can see we detect many more loci on chromosome 11 and chromosome 12. And even we can confirm individual loci by compare a pair of the lines. They are highly related. For example, these 4 integration lines are actually from the same BC1F2 plants. They're selected for salinity. Then again, they're selected for second round confirmation. You can see that they are derived from the same BC1F3 progeny. You can see they differ tremendously the following segregation and selection. You can easily identify these loci associated with these 2 lines have resistant to all the 15 races, as high level resistant to all the races. These two lines differ. They have resistant to 3 of the 15 races. You can find the 2 loci associated with the resistance. This is a single locus associated with the immunity. So in summary, we found a total number of 62 loci, genomic regions, that are associated with the resistance to 4 XS races detected in 6 populations. Most about the resistant loci were detected specifically in a single population, were specifically effective against a single race. Of these 62 loci, 21 was effective against the weak, and the 36 were effective against the strong or more violent resistant races, and the remaining 5 are effective against more than both groups. We found the detected the resistant loci vary considerably in their effect, because we found 2 separate sets, including 7 loci into this parent lines, and another 4 loci from this parent associated with the hypersensitive reaction. We found 7 loci of a large effect. Lesion is more active effect is more than 10 centimeters, mapped to the chromosome 4, 11, and 12. 6 other loci of large effect on the chromosome 3 and 11. So on average, sequencing allows us to have a good resolution of the detected loci. If you look at it, on average, 60 loci have an average size of 1.5 millibases. But if we remove the 2 large segments, we found the average size is 1.2 megabase. That means this size of detection allows us to, by informatic search, we can easily identify the candidate resistant genes in this region can be verified relatively easily. Finally, here we have a lot. Both cases are incomplete activities. We have a lot of ongoing activity, including more comprehensive analysis that are being done to detect interactions association between the unlinked resistant loci because the number is large, detailed bioinformatics search for candidate genes in the regions of the mapped regions. We are also conduct the genetic and molecular verification of these key resistant loci and using different genomic tools. We are also sequence the 15 the bacterial blood races, so we are now analyzing data, trying to find the interaction of the polymorphism between the pathogen and the host. And also we are conducting systematic of large number of trait-specific interaction lines for functional genomic research complex set. This is something for our future breeding strategy. We want to create the largest genomic database for the rice core collection. In the end, we try to integrate our breeding activity with the genomic research. So a lot of people contributing to the work I presented. My group at CAS, the BGI group, this ERI group led by the Ken McNally, and also the BGI group led by Genyun Zhang, and they did all the sequencing data analysis. My groups do all this work in the bacterial blood resistance. The funding actually from the Chinese Ministry of Science and Technology, from the Bill and Melinda Gates Foundation, from the Shenzhen Municipal Government, from the Chinese Ministry of Agriculture, and the National Science Foundation of China. Thank you for your attention.
Thank you very much, Zhikang. So because of time limitation, we have time for one question. Yes, Jianzhong.
Thank you, Zhikang. I'm Jianzhong from Japan. I have two questions. One, could you tell us how deeply you will sequence your 3K rice? And the second, it's easy to use it population, maybe it's easy to mapping or to analyze QTA genes with major effects. How effectively do you think your system will be, to analyze the QTA with minor effects?
Okay, for the sequencing, this is whatever has been done is on average you have 13x on average. But certainly they are not evenly distributed. I can tell you that for certain, this set are including all the parental lines in our breeding program. Those lines have been sequenced in the greater depths with more than 20. For the germplasm sessions, we just sequence on the average, so you can see, but we are not going to do any further sequencing. But through the breeding, you see the breeding by shallow sequencing of these progeny lines, we will fill the gaps and for the gene discovery and for the larger scale through the breeding activity. So we are not going to do any further. That's all we've done. Regarding to the gene discovery part, I did not show the strategy of how the bacterial blight, we give example that the highly heritable trait, which is non-targeted because the select lines have not been selected for the bacterial blight resistance. But those lines have been selected for drought, salinity, submergence, yield, and all this trait, we call it targeted trait. We have a better strategy to find genes QTLs for the targeted trait is much more effective. One thing I can tell you, the number of the loci involved for each trait, because breeding is easy. We can easily divide the lines, but genetics is very complex. It's not, if you think the large effect QTLs can be easier, if it's large effect, it's always detected. But in these cases, we'll find many loci so-called small effect. They are not so small, but they are together forming group which have very large effect. So I think the strategy we are using, one-on-one strategy, has to be reconsidered, particularly when we're addressing the complex trait.
Okay, please join me to thank Zhikang. Thank you.